past the hour. And maybe it's a good time to start. And the first item will be Francesca Bernini, who co-runs this channel with Jose Espinosa Carrasco. Francesca will introduce today's guest, James Reecy. So Francesca.
Thank you, Cedric. So it's a real pleasure for me to introduce today's speaker because it's Dr. James Reecy, a professor in the Department of Animal Science of the Iowa State University. And he also is the director of the Office of Biotechnology. And throughout his career, he made an outstanding contribution to the fields of animal genomics, functional genomics, but also bioinformatics, becoming a key reference for the livestock genomic community. His research has focused mainly on the understanding of the genetic and molecular basis of complex traits, and he had a strong emphasis on QTL mapping, transcriptomics, and genotype to phenotype predictions. One of his major contributions has been the creation and the development of a widely used resource, such as the Animal QTL Database, which is personally something that I really use a lot, but also the Vertebrate Trait Ontology. Both tools have become essential for integrating and comparing data across species. He also had a central role in the FANG community, pushing forward the functional annotation of livestock genomes. His work stands out not only for its scientific impact, of course, but also for his strong commitment to building a community-driven, accessible resources. So we are really excited to have you today with us, James. I'm sure your talk will spark some great discussion, and maybe I will learn something new from you. So, James, the floor is yours.
Francesca, thank you very much for a wonderful introduction.
Thanks.
I'll hit share here. And there. So I was going really to preface this whole talk into what is in a name in terms of how is it that we name traits, genes, and other things, and the knowledge in which it brings. To start, excuse me, I need it. Oh, wow. I need to thank a lot of people from the bovine genome annotation group for the FANG community that I'll be utilizing some of the data here. For background, as Francesca talk a little bit about the animal core QTLdb, the core DB, as well as some of the ontologies in which we've developed in order to tie the information together and how we have changed them over time. A little bit of the bovine FANG community, and then really get into more of a discussion for existing AI tools and curation, how we've been trying to speed up curation of data into the database, and then ultimately some visions and plans for the future. Everything which I'm going to talk about initially in terms of doing this, really all of the credit should go to James Coltis, Chi Lee, who are wonderful collaborators over the years, Carrie Park, who has curated most of the data into the QTLdb as well as CoreDB, and then Juliang Hu, who has administered animal-genome.org for many years. And they're beginning to work more closely with MaizeGDB and Soybase in some of the things in which it is that they're moving things forward in. So one piece of the puzzle in which I wanted to talk about was really this issue of curation. How do we get the data that scientists have been extremely successful at publishing, and that publication rate only seems to be going up. Many of you probably get asked to review multiple publications a week, which is more than many of us can do. But really, there's more than 1.5 million papers a year going into PubMed, and how do we try to pull that information in more effectively. For the QTLdb, we've focused predominantly on GWAS studies. In the early years, it was all linkage for QTL, but that data now has been swamped by what is available for genome-wide associations. But then we're also trying to pull in eQTL, MQTL, EpiQTL, SPQ, AS. The list goes on and on and on. The difference would be is that many of these traits or many of these QTL are not necessarily tied directly to a trait, and there is a need in order to try to do that for the future. And then what are the other studies for the future that we need to try to pull together as well? When I say there's no shortage of QTL data, and Francesca mentioned that she utilizes it quite readily, this is the growth over the time that we've had the database, and it was really in about the 2014-15 when things dramatically increased in time. While this shows a decrease in the number, this is really because we removed the old linkage QTL between 23 and 24, such that everything, it's still available. You can find it, but it is not such that it has a prominent role that it had before in the database. So there is a slight decrease in that respect. In addition, the increase in trait heritability. Now we're headed up to about 7,000 total heritabilities that have been curated, and correlations for either phenotypic or genetic correlations have now topped over 40,000 that we have in the database in total. For those correlation data, you can find it either in a table form, and this is an old view because it's halfway understandable. This is when there were really only a couple thousand in there. More than likely that you would look at it now more in the context of this graph, trying to show the correlations. And in many cases, we have multiple correlations between, say, yield grade and back fat and other activities that are here for looking at the data. The two databases themselves for QTLdb and CoreDB are tied together by 4 different ontologies, and I'll talk a little bit about them in the future in the talk. And what they provide, though, is the way to connect association data as well as heritabilities and correlations in order to investigate whether it's pleiotropy or genetic synergies or even antagonisms that may be present in the species in which it is that we're so very interested in. If you go back in time, this is one of the first Wordles for the QTL database, back when it was predominantly milk yield and milk protein and milk percentage fat that was in the database. Today, it is a lot more complicated in the number of traits that people have done. And even this is a very simplistic showing of the overall increase in the traits in which it is that we have. And this goes really to the heart of what is in the name of, in this case, for a trait. And I'm going to look at just variation that exists in back fat. And by no means is this all of the back fat, just some of, and it's meant to be illustrative. That you have portions for thickness at a different location. So many different portions of back fat. One of the things in which researchers are really good at is generating new names for, in reality, very much the same trait that was there. In the case that is here, we have different methods where people used a ruler or ultrasound or even fatometer. But again, it was always back fat thickness that they were after. There's also lots of variation of time. And this time component in even the method is extremely important because all of us have seen that there are QTL that will exist at, say, week 14, but not be present at week 26. that is taking place. So things change over time. And then there are plenty of different locations that people may have done these traits as well. And the list goes on and on. But all of these end up being modifiers or variants on back fat thickness in many respects. And this has been a learning curve for us over the years. So the vertebrate trait ontology is what it is that we use today. You can download it and use it from many different locations. And this is work that we're doing with the Mouse Genome Informatics Group at Jackson Lab, as well as the rat GDB folks at Medical College of Wisconsin. Trait variants that we have. So we have moved from trying to do this, where we have traits on every different modification in which it is that you have, to try and do now where you have what we would call a base trait. In this case, body weight is a base. You can measure the body weight many different ways. You can measure it at many different times in terms of doing this. And then we have trait variants on that, or things that, in this case, for body length at birth, or fat subcutaneous fat thickness at the third lumbar. So it's these trait variants or the other locations. And what we tried to break them down is here for whether it was analysis that you had done or anatomy location or the environment heard. These are all different modifiers that we have seen over time. And this has given you a snapshot of what it is that we put into the database itself. So effectively, what we've moved to is you have sibling traits, where in the past we had trait A and it was QTL A, and then it was modifier A, B, C, D, but we would take all of those names and all of those modifiers themselves. What we've moved to really now is modifiers and quantifiers that are tied to this. So you have a trait, in this case, it could be back thickness, the different experiments that you have, but now it's the modifier and quantifiers that are tied to that. So we begin to have back fat thickness by a ruler at the first rib. And we try to break that out. So you can find QTL in this case for a base trait across all of the experiments, but then you will also find modifiers and then the quantifiers associated with those. So you could break it out and say, here are all of the things that have been measured by a ruler and all of the traits that may ensue. Or here's a trait and all of the different places it's been modified and others for measurements as well. And this effectively begins to take all of the information that I was showing before, and this is just blowing it up to where we now have in the black each of the base traits that we have, and then in the blue will be some modifier trait that is physically associated with that base trait itself. And this looks into the curation tools that we have in order to try to collect this information that we can go multiple levels deep. And we've tried to develop a process whereby we will do this in the same order each time across each of the species. And this has had a profound impact on the number of traits in which it is that you will find. So in the past, and this is prior to 2022, there were 2,000, almost 300 traits that we had QTL in association. By moving to the base traits, we've taken this down to only 418 traits in total. So we reduced it by over 80%. So all of these traits are in the base traits, but there will be modifiers to come back to get you to this actual number of the information that is there. So you can see some of the change that we've had over time. And hopefully this has been helpful for folks. In this process, and the speed up of association data that has been published, we've recently been working on trying to develop AI tools in order to make it more efficient, in order to curate the data. Because in the past, we had to do most of the reading and interpretation ourselves, and then input it directly into the database itself. The QTL database itself is a multi-stage process. And what we've now developed are some AI tools in order to streamline this. These are not ones in which it is that you will see yourselves. All of the things we have developed are freely available for everybody. And the impact on this is really in filtering publications that we need to add to the QTLdb as well as CoreDB itself. We had a test set where there was essentially 1,000 abstracts in which it is that we were starting with. And it took, in reality, a curator 18 hours in order to work through all of these abstracts to say, yeah, we know that we can do a really good job with the information that is there. So in essence, we've been able to process 65 times the speed in which it is that we were before. This is a listing of the different methods that we've used. And most of this work here that I'm showing is the work of Chi Li, an extremely talented computer scientist that we have in computer science at Iowa State. And ultimately, we ended up in the CUPUL-ST, and that's where we had that 98% recall. The precision was a bit lower, but the F1 was actually quite high. It was not the highest F1 and it was not the highest precision, but it does give us the highest percent recall and is even for the strict metrics when we put them together as well. Ultimately, depending upon the encoders in which it is that we were using as well, and whether or not you had the VT, the livestock product trait, or the CMO, you can see that it still ends up having high recall associated with this as well. So the tool that is there has dramatically sped up what it is that we're able to do. And we're able to do that on the vertebrate trait ontology that I was talking about before. But then we also have the livestock product trait. In this case, this would be measurements on meat or eggs or milk after it has left the animal and or ceases to be the animal itself that we're measuring. Then we also have clinical measurements. In this case, say a hormone level that you are doing, that it's not necessarily a animal trait itself, but something in which it is that we can measure. And then the fourth ontology that we're using is the livestock breed ontology as well, in order to try to bring different breeds together if we can capture that information. I'm going to switch gears now, almost entirely, in order to talk about a different piece of the puzzle that we're trying to pull together. In this case, this is data from the bovine fang project in the States. And this is a listing of the 47 different tissues in which we had. They were both from adult as well as from fetal, and they were from the sequence cow and offspring when there were parent relationships with the offspring. We had a lot of data in that we had roughly 100, or not quite 100 million reads for the different RNA-seq, and then roughly 25 million reads when it came to microRNA. This data has all been published. While for what it is that we had, we didn't really generate any more genes than what there were for NCBI. But at the time, what we had done is, in essence, more than doubled the number of transcripts that were available versus NCBI, and more than doubled the number of transcripts per gene on average as well. At that point in time, interesting, the number of non-coding genes or transcripts that we had was roughly similar to those that were protein coding. And of the non-coding ones, vast majority of them were long non-coding RNAs, and most of them were in the antisense or in intergenic regions. The other large category were the nonsense mediated decay transcripts that we were able to identify. One of the questions in which we took this data, and this is trying to pull it back to the QTL and the trait information, is to say that are there QTL and genes that are associated with each other by more than random chance? So we took the genes that were close to QTL and then the traits of the QTL and then all QTL and did a simple Fisher's exact test in order to say, are there genes tied to different traits? And ultimately, we had 2,000, almost 700 genes that were enriched for QTL of different traits like twinning or calf size, bovine tuberculosis susceptibility, or even residual feed intake. And we're able to find those genes. Interestingly, in a group of testis-specific genes, they were represented for different traits, and not by just a little bit. I mean, this is a Bonferroni adjusted p-value of to the minus 25 for body weight and others. But thinking about it for testis, all of this list in many respects makes great sense in that they would be potentially tied directly to those traits. And this is the number of genes that were associated with a given trait and the association that was there. The other, and this is really kind of an interesting experiment to look at, we had testis genes that had signaling peptides. So they should be secreted from the cell, in this case from the testis. And then we had a large number of pituitary genes that were close to QTL for normal sperm. And what we're showing here is the correlation of the genes in this case, that we have at least two different sets and looking at the genes themselves. And when you look at them, those genes the pituitary genes were an overrepresentation of NF-kappa B transcription factor. Well, the testis, and this is one which I don't fully understand, were more ubiquitin-independent protein catalytic processes. So maybe it's processing something external to the testis, which then transfers to the pituitary in order to change gene expression there. Similarly, you can go the opposite direction as well. There are pituitary genes that also had signaling peptides that were close to the normal sperm. And then here were the testis genes that were close to the percentage of normal sperm as well. And again, they were overrepresented for either Rab protein signaling or ubiquitin-dependent protein catabolic processes. So in both cases, those genes with the signaling peptides were somehow in a breakdown process. Don't know, again, as I said, what the real meaning behind that is, but it is very interesting that you can pull these different groups of genes together, and it looks like there could be a signaling axis between the testis as well as the pituitary, and that it's going in both directions, not just unilateral. We also then took this same data, and apologize, you can't make out the traits that are here, but we wanted to say for a given trait that we had in the QTL database, is it related to a different trait. In this case, we took, for every association, we identified the nearest gene to that association, which we realize that there is error that will be there because the closest gene is not necessarily what that uh, association is impacting. But we had a list of genes then for trait A and the genes for trait B, looked at the association, and then ran the Fisher's exact test as we had done before for genes and QTL. In this case, we assembled a trait association for all of the different traits and their association with one another. And it looks like this is very good because if you take a component trait like residual feed intake, which has feed intake and body weight and other pieces, they all come together in this similarity network. So it looks like you can reuse all of the association data that we have there in order to find relationships from trait to trait. So if you start a experiment where you're interested in a trait like uh, corpus luteum number or something like that, does this point to you may also want to measure these other associated traits with that because there may be an impact there as well. And this becomes very helpful as you are selecting on any given trait in terms of moving it forward. You can find this matrix where you can zoom into it in the viewer at animal-genome.org for the future if you're interested in looking at it. So we've begun to try to assemble some of the pieces. Here's pieces from FANG. Here are portions for the QTL database using the ontologies and other methods in order to try to pull that information together. What we're really headed for now, and this is where I have to give all of the credit to James Coltis and Chi Lee. They're moving this forward as the project themselves. I'm there really more just for crazy ideas in order for them to try to say what could actually be done. But can you develop AI-guided, in this case, knowledge graphs in order to facilitate this data integration and new genetic discoveries? We need to find new ways to pull the data together and visualize it in order to look at what is possible for the future itself. So one of the things in which they've started to do is for the QTL database, and then all of the ontologies as well as the core DB take all of this information and begin to put it together in a knowledge graph. And I'll try to work through an example. And this is just purely illustrative. But dogs is a animal, as is cows, and they are all living things. Cows, in theory, could eat herbs or plants themselves, or herbs is a plant, which is a living thing. But can we begin to pull together a knowledge graph, which has been done to some extent uh, in other systems as well, and then add in the gene ontologies, maybe gene expression data sets, and ultimately then develop a large language model interface in order to test this functionality. And they have begun to try to pull together the different information which it is that we'd have. So again, I'll try to step through this portion. So on chromosome 1, there are a series of SNPs in this case that are associated with residual feed intake. In the same location, you have this gene, which may have other associations, other synonyms, other orthologs, other associations. And another gene down here as well. Are they potentially now candidate genes for regulation for residual feed intake, which is related to the different traits that are here as well? And oh, by the way, you have other SNPs that are associated with these traits as well. So can we begin to take the different traits, an entity in this case, or a different property associated with it, and then the genome identity information as well, and begin to tie it together into the knowledge base in order to make more predictions for the future for genetics. This information and what's on these edges is probably that information which is extremely important in order to try to pull together in a large integrated fashion in order to say, here is why we are getting the genetic correlation or even the phenotypic correlation that we're getting between the different traits because of the different associations in which it is that we have. So how can we pull this information together in order to identify more information for the future? Um, Ultimately, hopefully, what I'm trying to get to is what is in a name in this case is that we begin to take the data in which it is that scientists have been really prolific at generating, utilize the names, in this case for the traits, for the genes, for the SNPs themselves, and use that as the interconnectors between each of these that hopefully we can begin to develop a model whereby we can move towards prediction of the impacts on traits, not just predictions of here's from a genetic standpoint, but prediction of the genes, the SNPs, and the variants, and all of it together in order to explain variation within traits to a greater extent than what we could in the past. I need to thank USDA NIFA. They funded the majority of the work for the QTL database. I did forget actually to put in the NIFA number for the award for the FANG project. And then Zoetis has been very helpful in providing lots of information for us as well. So at that point, I think I will stop sharing. And if there happen to be questions that people may have and ideas that they have for the future of how all of this information could come together in order to help people to do the great things in which it is that you're doing.
Thank you so much, James. This was really a great talk, and I'm sure that if you could, everybody will be clapping in their hands. So I will now moderate the question and answers. If you have a question, just unmute yourself and ask directly, and this is usually civilized enough so that we can do this. And while we wait for this, I have a very, very generic question here. You know, hearing you talk, we are very exposed to human genetics in my institute because we host the European Genome Phenome Archive. And if you look at human genetics, it looks like this is only about sickness and about poor traits. And that seems to be amazingly complementary to the genetics, the cow genetics, which is mostly about healthy tissue and describing healthy tissue. How do you foresee the relation between these two genetics in the future? Are they going to keep helping one another? Are there some aspects where cow genetics is really driving human genetics?
So interesting question, Cedric, and I'll go in a slightly different tangent to start with and then try and come back to your question. One of the things in which I would look at from everything that we have in the QTLdb and even in core, it's based upon really quantitative genetics only. We have not really tried to start to pull in all of the evolutionary aspects that are there. So what if you ended up now collecting trait information on all species, all animals, all the way down to mosquitoes, up to cattle and everything else in terms of doing it, and try to pull that information together. So it's that we now have both quantitative genetics as well as evolutionary genetics in order to explain variation in traits. And even within our livestock species, I think there's lots of evolutionary information that could be captured there that we're really just starting to get to. Martine Gronin was doing some of that work with the pig genomes in which it is that it was sequencing, as well as now the pan-genome projects that are ongoing. Take that together, now bringing it back to the health data that you had from the human standpoint. I think the answer is yes. And in many respects, what you have are the livestock species, or any animal species for this matter, becomes the, I guess, test organism in order to say, under a healthy standpoint, this is what the genetic mechanism would look like or should look like. And now on the human side, all of a sudden you've got dramatic variation from that. And when you deviate, is that the place in which it is that you should be looking when it comes to those traits of interest? Because it has changed. Uh, what you would consider to be more of a healthy animal together. I would hope at some point in time we are able to integrate. And that's where that knowledge graph where James and Chi are headed to, I think has that opportunity to do it. And I also believe there will be opportunities to take it even across plant and animal in some respects as well. Now, every time you start get more evolutionary diverged, it's going to get harder in order to do that. But some of those same genetic mechanisms are more than likely going to take place as well. And those would be extremely interesting because they've been evolutionary conserved for so long. So hopefully it gets to your question in some respects, Cedric.
Yes, you do, and it's very, somehow seeing your use of LLMs and all these kind of things, I have the hope that at some point we will have a sort of meta traits collection that will
Mhm.
describe, combine all of these traits exquisitely analyzed in one species and not so well analyzed in another species, but this information
Mhm.
can come across. We have plenty of
Yeah.
questions. I saw Brenda's hand up and then
Yep.
Sylvain. Brenda, please unmute yourself and go ahead.
Thank you. Great talk, Jim. Awesome. Glad to hear what's an update of what's been going on. I have two questions. The first question is, you're only looking at abstracts for your AI.
That's what we did for the original test in order to make it very simple. We now are essentially using the same AI in order to try and pull information directly out of the manuscript itself, which has been challenging because PDFs are not all the same, and trying to get information out in a more automated fashion has been challenging. But abstracts are the one thing that are really quite simple.
Okay, great. Because yeah, a lot of stuff doesn't make it in the abstract, and I'm
Right.
sure that the actual manuscript is much richer. So great. Glad to hear you're doing that. So then my next question is, how is there, you've talked about a lot of traits and a lot of things, but I'm wondering if there's an opportunity to capitalize on additional environmental data, like
Mhm.
where were the studies, and have you thought about that and how that might be incorporated?
Great question, Brenda. And actually, that is a very tricky question because, for the most part, in all of our publications that we have, most people never say anything about the environment whatsoever where the experiment was done. What were the location of the cattle? Were they in Brazil? Were they in Argentina? Was it in the summertime? Was it in the wintertime? Most people tend never to put that information into the manuscript. Not, but unlike that, I would say our nutrition colleagues almost always put that type of information directly in their manuscripts. So there is an opportunity there, and there is an environmental ontology that does exist. We've not tried to pull it in yet, Brenda, for everything in which it is that we're doing, but it is something that may be possible.
Great, thank you. I think, as you mentioned, they are opportunities as we really try and understand how environment plays a role with these different breeds and predicting phenotype, I think. Thank you
Yeah,
very
I
much.
mean, you just simply look at cattle. I mean, the Holstein cow works wonderful in Europe. You move to Central Africa, not so much anymore. There's an environmental genotype interaction there, very true. Same thing for breeds of beef cattle, those that work okay at high altitudes, those that you just never move to altitude, those that are tropically adapted or not. There's lots of opportunities there, but unfortunately, much of that information is not collected.
Thank
Sylvain?
you. Thank you, James. So we now have a question from Sylvain. I will just want to mention on the abstract, as chief editor of NARCAB, I find it depressing to see how many scientists think an abstract is a teaser. We should tell them an abstract is not a teaser.
Yep. There is a method to the madness of what should be in an abstract. Absolutely.
Yes, yes, yes.
Yeah.
Sylvain, please unmute yourself.
Hello. Hi, James, and thanks for this nice presentation. These knowledge graphs are really fascinating. So I was wondering how easy or complicated or even possible is it to update them when a new version of genome assembly or genome annotation is available?
So unfortunately, I probably can't give you that answer, Sylvain, in terms of doing, because it's where my colleague Chi Li, she's the one who's taking the lead on all of that from a computational standpoint. I do know for what it is that we've assembled to this point, it has not been overly computationally burdening, but we're only in early stages, I would say at this point. So to say, have we integrated everything together? No. I mean, that's going to take probably more computational power than what it is that we've done to this point. But myself, I could not give you a good one, but I can put you in touch with Chi, Sylvain, and you can ask her directly, and she'd be able to give you a much better idea.
Sure, no problem. Thanks. And I was also wondering, this trait ontology. Of course, a lot of us are familiar with the gene ontology already, and so it's of course it's similar in the sense that conceptually trees and the hierarchical levels like this of organization are similar. And for gene ontology, not everything is in the same tree, if I can say like this. As you know, there are these categories, like general categories of biological process or cellular components, And I was wondering if you had to, come out with like general groups like that, or if you could manage to put everything in the same hierarchical structure.
So we're trying. Now, all the relationships that we've been using is ISA in terms of the relationship within the traits themselves. It's been relatively easier when we're limiting to what it is that we're doing for livestock. We have some collaborators who are now trying to take the vertebrate trait and move it to all animals using the base for which it is that we have. And there it has been more challenging, as for the very reasons in which it is that you're pointing out, that all of a sudden now you have multiple animals that they may have evolved traits differently. They may have subtle differences in trying to capture that portion. The other is, and this is an issue, if you go back now probably the better portion of 15, maybe almost 20 years ago, there was Pato, or is Pato, where you have an entity and a qualifier associated with it. And it was great for doing disease type traits, modifications, describing the body, but trying to capture the traits that most geneticists say within the livestock community were there. It was confusing, but in what we've evolved to for the base trait and the others, we really have decomposed these traits in order to try to make that more possible. The difference would be now is that this is not really just a simple ontology in terms of doing it. You could do the ontology very easy for the base traits, but then all of the modifiers, and this is where it now has to move to more of the knowledge graph, in that you have what was the location, what was the method, what was all of these other pieces in order to try to visualize the portions associated with it. But that's only possible if you condense it down to really that base trait concept. Otherwise, yeah, it just proliferates ad nauseum together. And it makes it very hard to begin to pull them all together.
Thanks.
We have another question from Trevor. Trevor, if you want to unmute yourself.
Thank you. That was a great talk. So I'm kind of interested in this AI knowledge graph that you're building. So I'm just kind of curious from an end user's perspective. I know this is still in development, but what's sort of the end goal for an end user? Is this like a graph rag that we would interact with through a natural language chatbot? Is the idea to post this as a database that we can directly query? Just kind of wondering what that looks like in your imagination.
I'm going to guess that your imagination is much better in this space than mine, Trevor, in terms of doing it. Because what the future will fully hold is yet to be determined within the space. But the pieces in which you have to say, can you query it? Absolutely. The intent is in order to have it in that space. Can you do things where here's the evolution in time for the information that we have, such that you can say, okay, all right, we brought in this data set and it changed this portion of the graph. Are there things there that you could glean from it? Are there ways that, as a chatbot, that you could try to pull from it? Because in many respects, you could say, could you write a query, give me back all of the list of SNPs that were associated with this trait, and this trait is correlated to this trait. What SNPs are there? And are those SNPs in this data set correlated with one another? And ask those types of questions. I would hope things like that for the future and many other queries are possible, but where it's all going to go and how much compute is it going to take in order to facilitate that? Because one of the things in which Chi is very interested in doing is trying to optimize the amount of compute it takes in order to do these pieces, just realizing that with every new data set that you keep adding, that the combinations just go up exponentially.
Yeah, that's always the issue with working with a graph, especially.
Yep.
Thanks for answering my question.
We have one more question by Praveen. Praveen, please unmute yourself.
Hi, James. Nice talk. I also have a similar question like Trevor. Using these LLM models, in the future, we would expect something like a cross-species queries, like even including plants or something like that. What is your opinion about this?
So that is one of the test cases that James and Chi are headed to right now. So that when I mentioned earlier Soybase and maize GDB, they're trying to tie the portions in which it is that they have from those databases directly to the QTLdb to see if there are some opportunities in that space. And they're running a project right now trying to do that. So I would hope the answer is yes. Now, going across plants and animals, probably as extreme as you could possibly get. Things that are going to be closer, say dairy cattle, beef cattle, probably much more informative in that space early on would probably be the easier pickings in terms of new knowledge that is gained. But the opportunity to do something across genus, I think, is really exciting for what may come in that space.
Thank you.
Thank you, James. We are going to the hour. I have one more question on this. So the more we look into these things, the more it looks like all of these LLMs will need to be fed all the time, continuously. And it's not something that you're not going to do once, drop here and say, job done. And as time goes by and the Earth's biogenome starts delivering and phenotypes accumulate all over, even in human genetics, the key word now is phenotype and traits, and how do you measure them? So what do you think of this infrastructure to come? Will it be something that is constantly rewinding itself? And if so, how will we stabilize it somehow? Because you cannot rewire everything every day. You need some kind of memory. So who is going to help finding the sweet spot in all these things?
Great question, Cedric, in terms of doing it. In many respects, I think we've already answered the question in some ways. This same question arose when we started annotating genomes 20 plus years ago. So this is not a new problem. What's new to it is the number of data sets and the degree to which you're integrating them and the computation associated with it. I think it's fairly easy that if the community or communities come together and work collaboratively to the extent to which it is that you can do releases on a timed component, whether that's a yearly time. Is this when a new data set comes available that was not there before, that is a leap forward? I think there are some go, no-go decisions in terms of doing that would be possible. But the biggest question in many respects is who's going to pay for that integration directly, because there will be knowledge associated with it, agreed. But the amount of compute in which it is that you have, is this something that now leaves what are maybe the European model in which it is that you have is better set up to do this than where we are currently in the States associated with trying to do this type of work? I don't know. But any time in which it is that we could work collaboratively internationally in order to do it, I think is going to be extremely helpful for the future. But as I said, I don't think we have to reinvent the wheel here. We've answered some of these questions in how we were handling projects in the past. The only difference is that N is a little bit bigger now than what it was back then. And we were afraid, I mean, back when we first started annotating genomes, that, oh, you can only have one assembly for a species in terms of doing this. And now the pan-genome projects have moved that past. So we've had solutions that have presented themselves because people now had an opportunity. Oh, I never really thought about that question. What can I do now in that space in order to solve what in the past was not a problem, but now becomes an opportunity for the future?
Thank you, James. I have to admit I had a little bit of a vested interest in this question because with Nextflow and NF Core, we have been very actively promoting a quite decentralized community, in the sense that many people can collaborate together on the same pipeline without having to require too much coordination. Why is it? Because the standards come along with powerful testing capacities, which mean that if you push your stuff on GitHub, if it breaks anything, it comes back to you and you have to figure out. And I have the impression that we could work out similar methodologies for all of these graph-based annotations. You know, we could tell whether some new addition is too disruptive or is breaking established things. And so there could be a very decentralized way of collectively working toward this. And the decentralized thing is interesting because it helps a little bit the funding in the sense that it's a sort of micro funding that is eventually adding up onto the resource. Everybody comes with what they have in hand. And talking about funding, Dominique Rocha says this is a great talk, and that he's asking whether USDA still has fundings to take care of the QTLdb.
Thank you for pointing it out because I had missed that there was something in the chat, Cedric. So right now, we have James has some funds from what is a community portion. So the old NRSP 8, if people remember that project, is providing some of the funding for the project. The work for the AI component of it was a proposal that we thought was going to get funded, but unfortunately, with the changes in funding in the states, that was not funded. They're resubmitting again here very shortly, and hopefully they will then have additional funds for continuing those aspects as well. So there are some portions of it that is there, Dominique, but by no means do we have everything to the level to what we did in the past. We're trying to build that pipeline back up.
Yeah, we have to brace for funding difficulties everywhere because there's no secret money. If they, in Europe, they are talking about, I think, 1,800 billions in, for the army industry. And so, yeah, this money has to come from somewhere. It's not going to be easy. It's something we have to realize. I don't see any more question in the chat. James, this was a really great talk, very inspirational. And what I especially liked is that it was a very powerful link between the 90s and the next decade. And, you can see EQTL and traits traveling through this period. So that really, really was a very nice piece of scientific history and very forward-looking, which is the kind of things we love to have on this channel. Thank you so much. Everybody joins me in thanking you. And Jose, I think you're going to close the session now because we are right at the hour.
I'm not. Yes, I thought I was not unmuted. No, it's a pleasure for us to announce that for June's meeting of the NF-Core Animal Genomics Special Interest Group, we'll have Angela Canovas, a professor in the Department of Animal Bioscience at the University of Guelph in Canada. Angela's research is focused on the integration of quantitative genetics, functional genomics, and bioinformatics to unravel the genetic mechanisms underlying complex traits in ruminants. So if you are interested, you are very welcome to join. The meeting will take place on the 8th of June at 4 Central European Summer Time. I think we are still struggling with the time shift because many of you have just joined. But I would like to remember you that if you cannot attend the meetings or if you want to revisit them, they will remain available on the NF-Core YouTube account and listed on the animal genomics sites on the NF-Core website. And just finishing mentioning that James' very nice talk was attended by 32 people. And thanks all for joining, and we are looking forward to seeing you next month. Oh, thank you very much.
Your camera is closed, eh? If you want to wave goodbye. You're very mysterious.
Because I don't know why my camera changed from the laptop one to the other one. Yes, sorry. But yes, so thank you and see you next month.
And see you next month then. Bye bye.